If you look at it, there hasn't been a major innovation in the PCR cleanup plasmid purification part of the workflow since 1990s when column was invented, which is a very labor-intensive and time-consuming part of the process.
I spent a lot of time in school, in previous jobs, doing cleanups, doing purifications, and it gets really repetitive. It eats up a lot of your day. And once you start doing hundreds of them, that time adds up.
You're looking at 40 to 45 minutes of wasted time. It's a coffee break. It's a chance to go and sit at your computer, write up experiments. It's not much fun.
It's not really rewarding, but it's something that has to be done to move things forward and do some testing of whatever you're making in the process.
The EMnetik system is really exciting because it makes DNA cleanup easy. So it takes all of the guesswork out of DNA cleanup by removing some of these barriers that you may face with different technologies and just making the process simple. For example, we have a user interface on the instrument that will actually walk you through each of the steps, and the steps have been simplified and there are only a few of them. So it really makes the process very user-friendly, all while producing high-quality results and in less time compared to other technologies on the market.
It's almost 2.5 times faster than the traditional PCR cleanup methods and almost 6 times lesser touch points.
From a lab perspective, using the EMnetik gets rid of your user variation, really. Using the magnets instead of having people manually move back and forth between a column or between beads on a manual setting means that user performance is going to look pretty much the same from one to another.
So you can forget about walking back and forth to that centrifuge and transferring your samples from tube to tube and really losing track of which sample is where, because that's no longer needed with the EMnetik 24 system.
After running it and seeing how I could do 24 samples in around 16 minutes, it was just, a no-brainer for me that this is an advancement in what I was doing prior to now.
I look at the EMnetik as a really unique thing because it takes a lot of what Beckman Coulter does from an automation perspective and makes it more benchtop friendly. We're looking for a potential user who maybe doesn't need to do 1,000 preps in a day, but still wants the benefit of the automation technology that Beckman Coulter has been developing. And so what I see is this kind of semi-automated thing gives people some of the advantages of the automated thing without needing to go full scale to that degree.